So I'm Lenny Rome. I have two jobs here. I'm the research dean in the medical school, but I'm also a professor of biological chemistry, so I have the same problem that you all have, and that is I have to keep my lab running and stay funded, and it ain't easy. So, but I have more gray hair than anybody else in the audience, so I've been here longer, and so I do have some advice for you if you're interested in grant writing and what to do about getting money. And so I've tailored this lecture for anyone who's interested in writing grants to the NIH. So, but you'll see throughout in the middle of the lecture, I'm also going to recommend that if you're interested in the kinds of things that I'm talking about, we've videotaped yet another lecture that I think is quite good that I'll talk about later as well. So we can tie to that lecture. So the name of my lecture is How to Prosper in 2012 and Beyond. I want to start with what I call the hard reality. 10 to 15 years ago, one could expect to apply for your first major grant from the NIH after completing a postdoc. Today, the average age for someone to receive their first R01 grant is over age 40. So that's pretty amazing. In 2005, 15 to 18% of grants were funded. We thought that was bad in 2005. Through most of my career, funding rates were in the 20 to 25 percentile. When I was a graduate student, I won't tell you how long ago that was, my thesis advisor, professor at University of Michigan, used to send money back at the end of the year when he had money left in his grants because he always knew that he could get as much money as he needed to do his research by just writing another grant. That's how it was. Okay, so amazing. For 2010, less than 10% were funded. Actually, this slide needed to be updated to 2011. It's the same. It's around the funding first time submission in 2011 is about 9.5%. For the past 6 to 7 years, the funding for NIH has been flat, which means that in considering inflation and in real dollars, the NIH budget is slowly going down. And if the automatic cuts come in that were triggered by the super committee not being able to come up with a budget resolution, if that happens, next year we expect NIH to have an automatic cut of 11%. So the funding rates will go down even more. So how do you get started? So obviously, this is a tough thing. I think it's important that you understand what is expected of you. Not all of you are assistant professors, or some of you have different departments that you're in. If you're a basic scientist, there's more responsibility on you to generate your research funding. If you're a clinical scientist, you have mixed responsibilities. So I would say for any of you on the web or here that are brand new, it's very important that you understand what is expected of you in your series and where you are in your career. You need to set realistic goals. Do not expect to get funded the first time around. And if you are submitting an R01, expect that you will have to resubmit. That's just the statistics. Explore your opportunities. What I mean by this is, if your primary research interest, okay, is, say, understanding the structure and function of the appendix, all right, you should go out and find out whether there are opportunities out there from the federal government or private places that fund research on exploring the structure and function of the appendix. And if there are not, get a new primary research interest, okay? I mean, I hate to say it, but you need to match your interest up with opportunities, or you are being unrealistic about your ability to get funded, okay? It isn't enough to be passionate about something unless you really are going to figure out a way to do it without money, all right? Okay? So I know it's an old joke, the guy who loses his contact lens and he's searching around, it's at night, he's searching around, somebody asks him what he's doing, he says he lost his contact lens and he's looking for it. And the guy says, well, let me help you. Where did it fall out? He says, over there. He says, well, why are you looking over here? He says, the light is better over here. Okay? So, okay. The problem is you need to be interested in an area where at least it's realistic that if you get preliminary results, if you have good ideas, that somebody is going to want to fund you. Okay? That's important. Get ready early. I cannot overemphasize this enough. You have to get ready early because it always takes longer to write a grant, even if it's not your first grant. If it's your first grant, it takes much longer. But it's even when I write a grant, I never estimate the amount of time properly. So it takes longer than you think. And secondly, you need time to show it to colleagues, mentors, friends, so they can tell you what's wrong with it and help you fix it before. You do not want your study section to be the ones that tell you what's wrong with it. Okay? So you need to take advantage of internal review. Very important. Okay. You have to be realistic about the cost of doing research. Not everybody is. In fact, it's amazing, but there are full professors here who've had grants before, but they really don't understand the financial costs. It's somebody in my department always makes my budget, they'll say, or I get help, whatever it is. I just want to point out that things are quite expensive now and getting more expensive. Personnel costs tend to be 60 to 85% of your costs. A simple lab assistant, college graduate, that you want to put on your grant is going to cost you $30,000 plus benefits. Okay? A staff research associate with a little experience is $35,000 or more. Or if you're doing clinical research, a research nurse can be over $100,000. Postdocs cost between $38,000 and $46,000. And guess what? Graduate students cost the same amount or more. Okay? Often you have to put a portion of your own salary or your co-PI salary on the grants. And what is often missed is the cost of benefits on top of the salary costs. And that's not something you can easily predict. If you hire a technician or a postdoc who has a large family and they need to put their family on their benefit costs go way up. And you're not allowed to determine who you hire based on what their benefits are. Okay? I mean, I suppose you could subtly find out when you're going to hire somebody if they're going to have a high benefit cost or a low, but you could never predict that. And I would just say, consider that when you write grants, you have to include 20 to 30% of benefit rates on top of salaries. Yes?
I'm just curious, do you know why the UC has this system rather than like what Cal State does, where everybody just dumps into a big pool and they...
I don't know. I didn't. In fact, I didn't know that Cal State had a pool. Here, it depends on what benefits you're absorbing. Yes.
So there that everybody pays a set. Each individual employee
Right.
pays a set.
Yeah. Might be a better system. It's probably something grandfathered into the UC system that we have this. As I mentioned, graduate students can be very expensive. The cost of supplies is high. Bench science is typically 20 to 40% of the total. Animals are incredibly expensive. The most expensive research that I do is any research that has to do with needing animals for it. I published one paper on a tumor induction in an animal model. The paper alone cost me $250,000 in terms of animal costs. It was ridiculous. Got to consider some equipment maintenance. If you're in clinical or social sciences, it's less for supplies. The physical plant and administrative costs are usually part of what you pay in so-called overhead. Then there's a cost for equipment, and there's a thing called shared instrumentation grants that we can talk about later. So I want to talk to you quickly about sources of funding. I do a whole introductory lecture during the introduction for new faculty about sources, but I'll just make it quick. The major sources of course, the federal government. Even though I was complaining about NIH, they still are where the really big bucks are. Even though everyone's complaining, they still have, in 2011, about $32 billion of total budget to spend. About 20-some billion of that is for extramural grants. The NSF has $7.4 billion. The American Cancer Society has $100 million. Then there's big societies as well, like the American Heart Association. There are many types of grants. I'm going to talk to you about them in a minute. There are other government sources of research grants. AHQR, this is Health Services Research, the Department of Defense, the Department of Energy, the Defense Department, DARPA. Has anybody heard of DARPA here? So this is the Defense Department's basic research area, and they will support research that is defense related. And if you're doing something, let's say, that has to do with health of military personnel, preventing them from getting certain types of exposures to nerve gases, things like that, it can be very relevant to biomedical research. There are large foundations, such as the Howard Hughes Medical Institute, the Keck Foundation. There are smaller foundations that are all usually disease-oriented, cystic fibrosis, multiple sclerosis, breast cancer, etc. Other useful types of funding include, specific to California, we have the California Institute of Regenerative Medicine here. If your research is related to stem cell research, human stem cell research preferably, then you can tap into $100 million a year that is specific to the state of California that was set by an initiative in this state. There is also specific money for research out of the tobacco initiative. Many states have this as well. There are training grants. There are industry-sponsored programs like the UC Discovery Program. There are private donors that people in clinical departments can often have ties to. And there are some small UCLA intramural programs. You can learn about these on the School of Medicine website. I want to take you through what I call the alphabet soup of award types because everyone's confused by this. I'll go relatively quickly. The most important one is the R01 grant, and I'm going to spend most of my time talking about R01s. But there are small research grants called R03s, which are difficult to get, but interesting. There are R15s that don't apply to you because they're for small institutions that have less than $6 million a year in funding, not UCLA. R21s are nice because they're exploratory grants that do not require any preliminary results. So I see full professors with other NIH R01s applying for R21s because it lets them open up a new area. And I see young people applying for them as well. Unfortunately, they have about the same success rate as the R01s. However, they have a different bar. So if you have a good idea and can write a good grant but don't have preliminary results, the R21 might be a good place to start. There's other things, BRP, engineering partnerships, R13 and U13, support for conferences, and there's a group of R41, 42, SBIR, STTR grants that are designated for small businesses and innovation awards having to do with businesses. These typically are one-time grants because you're interested in moving some product through the pipeline, okay? And often the type of audience I'm speaking to about this is not quite ready for developing a new drug for the pipeline. There are a lot of fellowship awards. These are called F awards. F30s are predoctoral fellowships. F31 are National Research Service Award individual predoctoral fellowships. There are F31s, there are F32s, and they're described on your handout on this slide. I don't have time to go into them. The K awards are important for this group because they are the mentored awards. They range from the K01, K02 down to the K99 here. I like this one up because it's a very important award for first-time faculty to be involved in. You can apply for a K award when you've just gotten an offer from a new institution. So when you're brand new, you should be thinking about the K award. It has a pathway for getting your first major grant as well. It's a very interesting award, and it has a higher probability of funding if you fit the categories for a K award. So you definitely, if you're new, you should definitely be looking at the K award and its requirements to see if you qualify for this type of an award. Other awards include the P awards. These are P01 is like an R01, but it's a big program project. It's like putting 3 or 4 up to 5 R01s together in one project. It's usually written by a very senior investigator. The reason I mention it is it's often a good opportunity for a young investigator to get their first grant by connecting their research, hooking their wagon up to a big, powerful wagon train, okay? So you got this big engine, this big investigator who's got a ongoing grant that is going to write a big program project, and if your project fits into it, the bar for you getting funded might be a little lower because you're going to ride the tide of this tidal wave of great research that this investigator has put together. So it can be a very good way for you to get your first grant by tying it to a successful research program. It's not so simple. You have to fit well into that program. The person who's writing the PO1 is taking a risk by putting in a new project that, if it doesn't fit in, could sink the whole P award. So it has to be a good fit. I'm not saying it's easy, but if you can have such an opportunity, you should definitely look into it. There are, I mentioned shared instrumentation grants. These are the S awards. There are institutional training grants. These are the T awards. There's a new type of award that starts with a U now, which is a research project cooperative agreement. It's a way for the NIH to take more control over grants by using this U mechanism, and it's usually a larger award than the individual research grant. Okay, so I just want you to understand the alphabet soup of all these award types. Okay, moving on. How do you get information? So I mentioned to you that there are plenty of websites where you can get more information about these things. I've listed some of them for you. The NIH website, the Cancer Research Organizing Committee, the University of California has websites. Sponsored research here at UCLA has websites. My office maintains a website including a search engine for you to search individual opportunities, both intramural and extramural. I told you about small research grants that are sponsored by the institution. Those can be very important because if you're going to need preliminary results to even apply for a grant, you're going to need a small amount of money, what we call a seed grant, in order to just get some preliminary results. We have a very nice program in the School of Medicine for medical school faculty to apply for $30,000 seed grants to allow you to get preliminary results. And we've shown great return of our $30,000 investments by investing them into faculty, allowing them to get some data, and showing that that has led to them getting their first grants. So if you're new here, you definitely want to take every advantage of not just startup money if you got that from your department, but any types of seed grants that you can get through the School of Medicine general funding or through specific funding like seed grants from the Cancer Center or the AIDS Institute or other places where they are more specifically geared to your research area. Okay. So I suggest that people think about having a mixed grant portfolio during their career. In the old days, it was like NIH. You know, if you were a major professor at a major university, you were funded by NIH, and that's all you worried about. That is not true today. Okay? Not only does NIH are they beginning to have salary restrictions on how much you can put of your salary on a grant, but the study sections, even though they're not supposed to, are actually looking at how many grants do these faculty have, and that is affecting their funding decision. So if your plan is to build a large research engine that's being sustained by a lot of grants, you should be thinking about diversifying your portfolio to allow you to mix up where you're going for your grants. I've had NSF grants, discovery grants, industry-sponsored grants, NIH grants, the whole mix. Specific agency grants as well, American Cancer Society. These are very important parts of building your career to do this. Okay, there are problems. Obviously, we're still in a recession, and private foundations, which were picking up a fairly good chunk of research funding, have cut back, but they still have money, and the stock market is rebounding, and they have to spend their endowments every year, so they do spend money on research. Should mention the Howard Hughes Medical Institute. They have a $14 billion endowment. They spend money to support what are called Howard Hughes investigators. So this turns out to now be an open competition. So if you turn out to be really hot, you can apply to become a Howard Hughes investigator. We have several here at UCLA. They get an average of, I think it's about $400,000 per year per investigator on average, so they do pretty well. Some of them have very big labs. They're still also competing for R01s and other grants on top of it. They do make a number of grants that are outside of grants to their investigators, so you should be aware that they have specific programs as well. I just list here, just for you to get an idea of the top 20 foundations and how much money they give and how many grants they award. And so, again, if you're in an area that one of these foundations is interested in, there is a real possibility that you can pursue private foundations for your research. Just to give you an idea of the current budget request that's into the NIH for 2012, $32 billion, as I mentioned, and it breaks down in the way shown here, but 52.9% of that is into the individual research grants, the R01s and that type of grant. I think something like 16,000 R01s will be funded in 2012. So this is by far the largest opportunity, but also the highest competition for grants. But money goes into other places as well, including a big chunk, 10% of the budget for intramural research at the NIH. Some of us think that's gross, okay? Well, if I can explain, if the NIH was a university, okay, if the NIH intramural program was a university, okay, I don't think they'd be rated in the top 5 universities in the United States. Yet they got 10% of the NIH research budget, okay? So I think there's a priority mismatch that has to do not with the fact that they don't deserve a lot of money, but for the fact that there isn't enough money in the NIH budget to support the large proportion of very good research that's going on in universities. That's the problem. NIH breaks down into their various ICs, institutes and centers, and the proportion that goes to the various institutes and centers is shown here, with the NCI, the National Cancer Institute, getting the largest chunk of the NIH budget. You can go through this later. So let me talk to you about the NIH grant process and get into the particulars of applying for grants and try to give you some advice in this area. The process is that you submit your grant proposal. It's all electronic now. You upload it to a website. It goes to the Center for Scientific Review that assigns it both to an institute and to an IRG, initial review group, which are these study sections that review your proposal and score it. Once it's scored, if it's in a potentially funding range, it still has to make it through an advisory council for a secondary review, and the council can make minor adjustments in where you are. If you're on the border, the advisory council can push you into the funding range. They can also push you out of the funding range. Then there's an administrative and financial review, and finally you're awarded the money. The grant proposal itself is directed to a panel of experts with diverse backgrounds and interests. 3 reviewers is all there will be that will get to do an in-depth review of your proposal. The proposal, therefore, must inform and persuade those 3 reviewers, okay? They have to be convinced that it's important for science, important for human health, feasible, that you are the guy or your team, you are the guy or gal or your team is the group that can do the work, and that your budget justifies what you're asking to do. The scoring now, it's a whole new system in the last few years. The scoring now goes in the following way. If the impact is high, the score can be 1, 2, or 3, and the descriptors are 1 is exceptional, 2 is outstanding, 3 is excellent. If you're in the middle, you can go from very good to satisfactory, and if you're rated low, you can go from fair to marginal to poor. There are also non-numerical scores. You can be NR, not recommended for further consideration. You can be ND, not discussed. This is a triage process. Somewhere between 1/2 and 1/3 of the grants that come into the study section are not discussed. They're just triaged. You don't even get a score. And you don't even know where you were in that 60% of the grants. Were you 41 or were you 99? Yes?
I was just going to point out about those that you do get
Yeah, you find out about how, yeah, yes, you get comments sometimes as well. You can figure out where you are. But the NIH, they're overwhelmed. The problem is they're overwhelmed by the number of good grants. And the system, the peer review system, in my opinion, was designed to be able to determine which 1/3 of the grants are superior grants, are the best work, science work, which 1/3 are not very good, and which 1/3 are in the middle. Peer review does a very good job of parsing grants into those thirds. And so peer review can determine with very good accuracy, I believe, the 33% that are in the top, okay? But when you get to funding levels that we have now that are less than 10%, peer review begins to break down because they can, in my opinion, peer review cannot do a very good job of taking the top third and determining which 30% of those are the best grants. That's the real problem we have right now. So the review criteria are listed here: significance, investigator, innovation, approach, and environment. And I'll just highlight them quickly because I have many more slides than we have minutes. So I'm going to run through some of this, and I'm going to give you ways to go back and backfill any extra information you're going to want from my presentation. This is a survey for you. Oh, sorry, and then overall impact. So first, the significance. Does the project address an important problem or a critical barrier to progress in the field? Very important, okay? In fact, you'll see, I think, that you need to start your whole process with an outline, not with a grant. And your outline, you'll see later, has to be subjected to a lot of scrutiny before you should ever sit down and actually write a grant. And a lot of it will have to do with what area you're in. Do you have the right preliminary results? Are you asking the right questions? And that can be done from an outline, okay? Also, if the aims of the project are achieved, how will scientific knowledge, technical capability, and/or clinical practice be improved? NIH sees grants that says this person's asking reasonable questions, they can do this work. And then somebody in the study section says, so what? If we have the answer to those questions, we're really not going to be taken forward into any kind of advance that makes any difference. You're dead in the water, okay? How will successful completion of the aims change the concepts, methods, technologies, treatments, services, or preventive innovations that drive this field? How is your grant going to make a difference in your area? Investigator. Easy for you guys because we hire very good people here at UCLA. Is the principal investigator or collaborators or other researchers well suited to the project? If they're early stage, do they have appropriate experience in training, mentoring they're going to look at on these K awards? If they're established investigators, have they demonstrated a record of accomplishments that have advanced their field? Okay. Not only what have you done before, what have you done for me lately? It's all there. If the project is collaborative, do the investigators have complementary and integrative experience? Is the leadership approach and governance and organizational structure appropriate? Innovation. Does the application challenge and seek to shift current research or clinical practice paradigms by utilizing novel theoretical concepts, approaches, methodologies, instrumentation, or interventions? Are the concepts, approaches, or methodologies, instrumentation, or interventions novel to the field or novel in a broad sense? Is a refinement, improvement, or a new application of theoretical concepts, approaches, and methodologies, instrumentation, interventions proposed? Okay, these are all parts of them wanting to know that you are putting in a grant that's innovative. Approach. Are the strategies, methods, analysis well-reasoned and appropriate to accomplish the aims? Are the problems, alternative strategies, and benchmarks for success presented? Not only do you need to present aims and how you're going to complete the aims, but you need to anticipate what problems you're going to have in the aims, and if you have those problems, what are you going to do to counter those problems if they occur? If the project is in the early stages of development, will the strategy establish some feasibility, and will a particularly risky aspect be managed well? In other words, here's a problem. If your first specific aim, okay, has the possibility of being answering no. And that all your other aims were built up on your first aim being yes, okay? You're done, okay? Right? So you cannot propose an aim that if you fail to accomplish it, it will inactivate aims 2 and 3, okay? So that's critical. That's why you need preliminary results, because often what you're doing in aim 1 is showing that aim 1 is almost done already. I've got all these preliminary results. Aim 1 is just about completed, and I know I can do aim 2 and 3 now, okay? That's why I say peer review breaks down, because when you look at the 30% top grants and then you're asked to pick out the top 10% or 1/3 of those, you tend to get more conservative, and you tend, as a study section, to fund that research where it's a sure thing. Okay? And that's not what NIH was set up for. If you're never going to fund anything risky, then we're never going to do anything creative. Okay? Nevertheless, that's the system we have, and so I'm advising you to put in a creative, wonderful, innovative grant that convinces the reviewers that you can and will do it. Okay? Approach. If the project involves clinical research, it's very important that you cross all your T's and dot all your I's with regard to protection of human subjects, inclusion of minorities and members of both sexes and genders, as well as inclusion of children if it's justified by the scientific goals and strategy. Okay? These are gotchas. If you don't do some of these things and you're in a clinical research area, they'll just toss your grant out without it being reviewed. Environment. Will the scientific environment in which the work be done contribute to the probability of success? You should spend some time bragging about the fact that the environment here at UCLA is very good for doing research. We have wonderful core facilities. You have wonderful mentors. You have great collaborators who are just down the block or down the floor from you, okay? This is a huge place. You have no excuse for not having all kinds of great connections with regard to our research engine. Our institutional support, equipment, and other physical resources available and adequate for the project. If you're going to use a core facility, for example, you need to spend time talking to the core facility director, maybe getting a letter saying, oh yeah, we can help you, it's a great project, we can help you, we have all the instrumentation, we have experience using this type of confocal microscope, no problem, okay? You don't want an issue to come up during the review. Well, this is an interesting grant. I don't think that investigator has any experience with confocal microscopy. I don't think they're going to have any way that they're going to be able to do aim 2, okay? You don't make that an issue. If you feel as though they could have that be a criticism, then plug that gap with a letter from somebody who knows what they're doing, saying to you, no problem. You'll be able to do that because I'm going to help you. Will the project benefit from unique features of the scientific environment, subject populations, or collaborative arrangements? Very important again for UCLA. If it's basic science, you can always find a resource here that is nearly unique, okay? If, for example, you're doing some structural work and you want to do some cryo reconstruction, you'll find out that we have the top cryo-EM facility in the world, probably here at UCLA. If you need special subject populations, Los Angeles is the most diverse city in the United States. There's a lot of community-based work being done here. You should be able to tap into that as part of your research and be able to brag about that to your study section so they realize that you are situated in an ideal place being here to do the work. Okay, overall impact. Will the project exert a sustained, powerful influence on the research field? The overall impact score, by the way, is not the average of all of the scores that you get from each of those sections I just talked about. But it is the score that determines whether you will be funded, okay? So that's the catch-22 here. Okay, common criticisms. If the project was successful, it's not likely to change the field. This is the so what, okay? If they do that, so what, okay? Or they missed important questions. It's not very innovative. This has already been done, or someone more qualified I know is already doing this. We funded a grant to this Howard Hughes investigator last week or something, and that guy was doing essentially what this young assistant professor wanted to do. and therefore they'll scoop them, and it's a waste of money. Methods and approaches are standard and do not push the field forward, so you got to be innovative. Common criticisms. Also feasibility. Again, if you're doing population-based research, you have to be very careful that you can recruit the subjects that you say you're going to be able to recruit, that the experimental power has been done. You need to consult with statisticians if you don't have the ability to do that. And again, we have very good statisticians at UCLA and at the RAND Institute, which we collaborate with readily. You don't want to get caught in that the amount of work is excessive for the resources or time available. This is the over-ambitious criticism, okay? Over-ambitious will kill you, okay? You have to be applying for, putting up aims and a budget that's doable in the amount of time you say you're going to do it. And then reagents and methods might not be up to task. The focus. You could say the work won't result in any coherent body of knowledge. It's unfocused. They don't have any contingency plans. There's no alternative outcomes considered in the plan. The whole project hinges on one part being successful. If that's true, that's fine. You can have a project where it all hinges on one part being successful. Show them already it's successful. Show them preliminary results. You can say, I got to repeat this a few times, but it looks great. And here's the results. I can convince you that it's going to be successful. All right, basic guidelines for successful grant writing. We've talked about some of this. I just want to bring it back again. Identify sources of funding. Be realistic. Is there places that are interested in your kind of research? Know what your funding source expects. Find out what their guidelines are. If it's not NIH, private places, etc. Know the requirements. Are you qualified? Is there a limitation of age, citizenship, time in your career? Do they require that you be young or experienced, etc.? Give yourself plenty of time. Make sure somebody else reviews it before you submit it. You really want to get somebody who will be tough on your grant, because really you only have 2 chances now, okay? You can only resubmit once now, okay? So you really want your first refusal to be your friends, okay? That's your first time to be shot down. And then you want your second refusal to be the first time you submitted it to NIH. Know your limits, your abilities, know when and where to ask for help. Now this part I highlighted, and I'm going to tell you only a little bit about this because this is a whole lecture that I don't have time to give, but fortunately we had an expert from NIH give this lecture last year. I'm going to point it out to you in a minute, okay? Learn who will be reviewing your grant. Influence this if you can. Know the process by which the grant submission, review, and notification occurs. In other words, you need to direct your proposal to the right review section, okay? You need to strategize within your proposal who the reviewers are going to be, okay? You actually have to figure out who are the 3 people that are most likely going to be assigned my grant in the study section, and then you need to write your grant to those 3 people, okay? It's amazing, okay? And I'm talking about to the extent of researching what their papers are about, what they've said in the field, so when there's any overlap with your grant, you're bragging about how good they are in your grant. I mean, it's so political that it's amazing, okay? Understand the scoring system, what you need to do. And so I want to point out that we hired Greg, we got Greg Millman, who was then a director of NIAID. He's now retired from NIH. He came last year and he presented a talk called Secrets of NIH Grant Applications. And there's some overlap with what I've been telling you. But there's a whole section in this lecture about how to do that political strategizing that I'm telling you about, okay? And it requires a whole section, and he does it better than I can do it. He takes an example area and walks you through step by step how you identify whether that area is appropriate, who might get it in the study section, and who you would write the grant to. I think that if I had heard Greg's lecture when I was a young assistant professor, I would have gotten many more grants over my career, okay? I'm not trying to set you up for this. This is really worth your while, okay? So it's in your handout, it's online. When this lecture is put online, we'll put a link to this lecture as well. We recorded Greg, and we've got it. Okay, so let's go through applying for your first grant. And in the last 20 minutes, I'll rush through this, but you'll have the slides. And I'll just give you some advice for assembling your first R01. And again, I'm going to go much too fast on purpose because you'll have the notes and you can go back and look at it. You can look at Greg's proposal as well. So you'll follow, if you're doing your first R01, you'll follow the standard form 424. It's all detailed on the NIH website, of which I give you the link. The parent announcement, these are all called funding opportunity announcements, or FOAs. The parent announcement, you can Google it as PA 11-260. It's the announcement from the NIH that describes how to do all these R01 grants, okay? And it describes all this stuff, the number of pages, the font, the spacing, the margins, how you submit electronically, the administrative information, the personnel, the biographical sketches, the resources, the research plan, etc. The meat of it is the research plan, so I'll spend most of the time talking to you about the research plan, okay? The research plan is broken down into the following areas: specific aims, significance, innovation, preliminary studies or progress, and approach. Specific aims. Market your application to the reviewers. This is everything that I've been talking to you about Greg's presentation. And again, I won't go into this. Choose specific aims that the reviewers can easily access. They should be achievable. They should achieve an endpoint. You should probably limit your application to one specific aim per year, okay? Most first R01s, I would recommend 3, not more than 4, even though you're going to ask for 5 years, okay? Focus, okay? Again, most new applicants are criticized for being too ambitious. Have no more than 1 or 2 sets of experiments for each aim. If you need more, you need to reorganize. Okay? Think of your specific aims as a framework of your application, sort of an expanded table of contents for your approach section. Make each aim a header. Design experiments to address each aim. Describe your specific aims as well-focused objectives to achieve. Describe options with milestones depending upon the results. Have aims that test your overall hypothesis. Specific aims itself is 1 page. Okay? It's really a longer version of your abstract of the grant. You could use them also, as I said, there's typically 3 to 4 numbered goals, 3 to 4 aims. Use them as subheadings for the research plan. Think of it as the outline of your proposal. Introduce them with simple 1 or 2 paragraphs that provide the rationale and background, and conclude with 1 or 2 paragraphs that describes the significance. Kind of think about the specific aims as a sandwich. Borrowed this from Tom Gantz. 1 page total. You got the rationale, significance, and hypothesis. That's 1 to 2 paragraphs. That's the top bun of your sandwich. You got 3 to 4 numbered aims. That's the meat and the cheese inside. And then you got this 1 to 2 concluding paragraphs. That's the bottom. Okay? That's kind of the sandwich. Significance. Frame the problem in its general significance. Briefly review relevant literature and preliminary data. Mention the data, the preliminary relevant data of the people who are going to be reviewing your grant. Right? Interpret and identify gaps in knowledge. Indicate in general terms how you will fill the gaps. Show how the work is new and unique. Assess the potential impact on science and health. Refer to NIH priorities as articulated in the request for proposals if you're responding to a particular request. Okay? This grant is in response to RFA number so-and-so, and the highest priority of that RFA is to do exactly what I'm doing here, and this is why. Describe how the work is new and unique. If it's highly innovative, you'll need to build a strong case for your ability to challenge the existing paradigm and your reasons for doing so. I'm reminded of Seymour Benzer, a former Caltech wonderful professor, who proposed to study neuronal circuitry by doing it in fruit flies, and his grant got trashed by the study section and said, you know, this guy's crazy. How can you study neuronal development in fruit flies? Of course, he was the whole pioneer of the field, and much of what we know has come out of his work, but he was ahead of his time. And so you would say it was Seymour's problem that he did not build a strong enough case to those reviewers that he could actually do this when no one else had ever done it before. So it's not that I don't want you to propose stuff that's highly innovative and that challenges the paradigm, but be prepared for the reviewers to not get it, to not understand your work, okay? And to not see the significance of it, okay? Being ahead of your time is just as bad as being behind, okay? Got to show the work's innovative, present a broad view of the field and its significance. Approach, a very key part. The sections of your approach are going to correspond to your specific aims. You can briefly reiterate the significance. You need to integrate your preliminary data into the rationale. You need to present the logical structure of the proposal. Use multiple approaches, but prioritize what you're going to do first. Some of those approaches you're going to do because if something doesn't work or if something goes this way, you're prepared. Discuss the pitfalls and alternatives. I've seen good grants that use a little section at the end of each of the AIM approaches where they say pitfalls and alternatives, and they go and they criticize themselves. Of course, they set up criticisms of themselves that are answerable, okay? They don't say this will never work and should never be funded. But in case this first idea, which is the most relevant one, doesn't work, I'm prepared with idea number 2, which is going to work, okay? You have to be ready to do that. Discuss the data analysis and interpretation if it's data-driven. Common or routine methodology can be lumped at the end. You do not nowadays have the time or space to put in much methods, okay? It used to be we would describe experimental protocols in our grants. We had 25 pages for this. Sorry, you can't do that anymore, okay? That's why it's important that young people have preliminary results, because in a way, a preliminary result also tells the reviewer that you know how to do the experiment, okay? And you didn't have to lay it out. Or you could say things like, and I've seen this commonly done, I've got a mentor or a collaborator who's published 10 papers in this area, who has all these as cookbook procedures running in the lab right now. I've already done half of them. There's no problem. You can say that to reviewers. You can reassure them that you know how to do these types of experiments, that you have the methods down pat, but don't spend any time going through them, okay? Don't do that. Flowcharts might be helpful. Provide your own key data supporting the rationale, data. Demonstrate relevant technical expertise, introduce the research team and its skills, point out special resources, as I said. So the layout then of the specific aims, again another sandwich here, is you have this brief background and significance, a hypothesis, a rationale, strategy and innovation all at the top, and then you sandwich your experiments in between, and then your anticipated results, pitfalls, significance, etc. Okay? Make this nice, neat, well-organized package. As soon as your reviewer gets confused by what you're doing, by your lack of organization, you're dead. Okay? You want the reviewer to finish reading your grant and say, it was a pleasure reading that grant. It was just crystal clear. I saw exactly where they were going. I think they can do the work. If they succeed, it will be significant. I like that. Okay? Boom. You're in good shape. Helpful hints. As I said already, use headings and subheadings. Break up the text with figures and charts. Make it appealing to the reader. Think about these guys, okay? They're sitting on a study section and they're not getting paid for it, okay? Their own research grants are pending or they're writing them. They're so busy. They get sent from their study section a box that contains 100 proposals, of which they're the primary reviewer on a bunch of them and the secondary reviewer on another bunch of them. So they have to read all those grants, okay? And so they're under stress and they pick up your grant and it's dense and it's uninteresting and you're at the borderline of whether the text is even big enough, okay? They hate it from the beginning, okay? It's a psychological thing here, okay? User-friendly. Make the reader Oh, this is a nice looking grant. Oh, there's a figure. That's what they're talking about. Take them by the hand, okay? You have to communicate excitement, but be realistic. You need to close with a future directions and a timeline, okay? You are very confident. You've just presented this wonderful proposal, and you also want them to know that you know where you're going to go from here if your proposal works, okay? So if you're going to cure cancer, you're never going to put it in the first grant, right? Because that's too overly ambitious, okay? But if your grant is going to lead to the curing of cancer, then you're going to describe that in this future directions. Having these aims in hand, I'll then be able to do X, Y, and Z, which will revolutionize the field, okay? Whatever, okay? Timeline is very important. They'd like to know that you've thought about what it's going to take to get to aim 2, to get to aim 3. It doesn't have to be a huge, long thing. Often I accomplish the timeline in an inch of space and a paragraph or two of description, because there ain't much room in these grants, okay? But you got to put it in there. More advice. I got lots of advice for you. Let's see which ones of these are worth spending any time on. We got a few more minutes. Be careful how much you propose. I already said that. Sharpen your application's focus. This is where I say go watch Greg Millman's advice about this. It is revolutionary, okay? Beware of being too innovative. As I told you, that could be a problem, okay? Not that it isn't great that you are so innovative that you are developing something that is going to revolutionize your field. Just be careful that you can convince your reviewers that you can do that, okay? Common reasons for failure. Goals aren't significant. Weren't any new or original ideas. You lack a demonstrated accomplishment relative to your stage. Preliminary results, except for the R21, and even with the R21, I see people putting in preliminary results. Except for the R21, you have to have preliminary results. That it was a weak scientific rationale. It was superficial or unfocused. You had a lack of understanding of the literature. You had inadequate methodology for your goals. You had a lack of experience that were essential in certain methods that you were doing. Illogical, disorganized, or presumptive approach, uncritical approach, scope excessive for the budget, lack of key experimental details. Must be provided very strategically in the new short format, okay? Like I said, you can't put in the details, but you got to convince them that you know how to do the experiments. Yes?
I would add to that, that doesn't have sufficient quantity of recent relevant publications.
Yeah. Well, if you're a young, new investigator, they can't ding you for that. They can't, I mean, they can do it, but they're not supposed to.
They did it all through the whole, like in every section.
Yeah,
it was like, great grant, nice science stuff.
That just sucks.
What was it? The exact wording was, resubmission strongly encouraged provided postdoctoral paper referred to in the text is published.
Yeah.
Just so everybody knows.
Everybody knows. So I do think that means that you are very promising. Your CV's a bit thin, and they're dinging you for it.
Yep.
That's what that means. Yep. Resubmission. Almost all of you are going to be resubmitting. Okay, just look at the numbers. Okay, I mean, in my entire career, okay, I've gotten lots of NIH grants. I've been here for 30 years. I've got lots. Okay, I believe maybe it's in the neighborhood of dozens. I think 1 or 2 I didn't resubmit. Okay, those were probably accidents. Okay, got one review back. Okay, in a 4th percentile, and I sent it around to my lab, and the comment that I wrote on it was that my mom was reviewer number 1. I mean, that was what my comment was. Okay, so you only get 1 resubmission now. In the old days, you could keep trying until you got it better. Subsequent proposals may be screened for similarity. You can't just change the title and try to get a 3rd resubmission. They'll get you. Introductory page is used for direct rebuttal and summary of changes. It's very important. When you are encouraged to resubmit, you need to grovel a little bit to the reviewers. Oh, you were so right, okay? The comments that you made have made me do a dramatic rewrite of this grant exactly the way you've proposed, and now I have addressed every single one of your criticisms and made major changes in those things that you were unhappy with, and this is where I did it, and et cetera, okay? will usually help. Except then you'll get screwed because it'll go to a study section that just rotated its members, and the guy that shot down your grant will not even be reviewing it this time, and the new person on the resubmission has a completely different set of criteria. So it's sad. Significance and innovation concerns are difficult to rebut, okay? When someone says this isn't innovative, that's pretty tough for you to rebut that, but you're going to have to give it a shot. Or it's not significant, you're going to have to give it a shot. Best rebuttal to criticisms of approach are new data and interim progress. Remember, you got 6 months to a year between when you got shot down and when your resubmission is in there. Hopefully you have a little seed money. You're operating on fumes in the lab or whatever, but you made some results, okay? So there's nothing better than a close grant that was worried about whether you could finish AIM-1, and now you've got AIM-1 finished when you come back in. Nothing better than that, okay? That would be great. So, I know I've been depressing here today, okay? So I said to my wife, she said, what are you doing tomorrow? I said, I'm giving a lecture to new faculty about getting NIH grants. And she said, looked at me and she said, are you going to tell them to shoot themselves? I mean, because she knows what I go through, okay? So I said, no, there's still a lot of money out there, okay? Yes, these are tough times. The competition for research dollars has never been greater, and the amount of funding from NIH is flat or going down, okay? Here's the positive part. There are still billions of dollars out there available for research funding. Billions with a B, okay? And a variety of sources, including the federal government, foundations, the state of California, and the university. This money will be spent, okay? They're not going to hold on to those billions. Every year, that $1 billion will be spent, billions. And those who are competitive will be funded. You're a faculty at one of the top research universities in the world, okay? Last time I checked, UCLA was number 4 in total research dollars among all universities in the United States, okay? So you are already in a good position to get this money, okay? You got here because you have what it takes to be successful, and I think by following my advice and Greg Millman's advice and your mentors, you will be successful because your mentors have been successful, okay? It isn't easy. It's harder now than it ever was before, but it's doable, okay? If there are any questions, I'm happy to take them. We still have a few more minutes before the web thing ends, but please.
You said your slides would be available?
Oh yeah, we're putting up the whole presentation. Oh, let me say, I want to acknowledge, I mentioned Tom Gantz, also John Adams has helped as well with certain ideas here. We've given this lecture in other forms. Our research website is here. The slides will be available. The videotape that Mark is taking here will be available. You can watch me again, watch me in slow motion, laugh at all my bad jokes all over again. It's okay, okay? But really, I do recommend that you watch. If you're only going to do one more due diligence, watch Greg Millman's presentation. He does a really nice job of talking about strategizing, okay? Other questions? Yes?
If you have any comments that are specific to people in the assistant researcher stage, that'd be...
Yeah, well, I think people starting out, okay, need to take advantage of some of the resources that are for new investigators. So as I mentioned, seed grants here, the NIH R99 mechanism if you qualify, the, I'm sorry, K99, the R21 if you don't have preliminary results. Those are things that you should concentrate on because you'll only be a junior investigator for a short time of your career, so you need to take advantage of certain resources that we are targeting to junior faculty. Our seed grant program here, we definitely skew toward young investigators, and that's also true, I believe, in some of the institutes like the AIDS Institute, Cancer Center, et cetera.
And is the medical school's seed grant program open to assistant researchers? AIDS Institute just opened theirs this last round.
Yeah, it depends. So if you're an assistant researcher, not yet in a faculty appointment at UCLA, you may be closed out from some of those seed resources because, again, we've designated that money to help our junior faculty become successful. And so in some cases, they were donor-driven funds, and those donors aren't interested in you being successful and then going and being successful at another university because they're UCLA donors. So that's part of the reason why we don't give it to people who don't have an actual faculty appointment.
So the rationale for AIDS Institute opening it was that assistant researchers are eligible for R01s.
Yes.
So we need seed
So,
money.
and that's a good rationale. And I think that if you have a pathway toward an appointment in your department, and you can convince most of the organizations around here that you're applying because there's a pathway for you to be an independent investigator here, it usually will allow them to make an exception and allow you to apply for seed funding. Yep. Any other questions? Okay, I'm happy to stick around. One more?
Is there any structure to the timing, like for different awards?
Yes.
Like I have-
So on the, definitely is. Greg goes through that in his talk a little bit. There are cycles of which all these occur, and you need to understand the cycles of submission, resubmission, where they are in the funding cycle as well. So there are nuances here that you need to understand.
I only asked because I did the February cycle for K.
Yes.
And I looked on there, and the meeting is not scheduled, the council meeting is not scheduled till October.
Yeah, well, that's probably because they only meet once a year. So,
So that was the.
yeah. So it'll depend on the award,
But
yes.
even for the R21, maybe that's something I didn't know until I did this, is that the whole thing, it takes a year.
Yeah.
So, and then,
Usually it takes a year because by the time you get your comments and your score back and then be able to resubmit, you usually don't even want to rush into a resubmission if it's a week because you really want to be able to adequately respond to the reviewers since you only have one shot on a resubmission.
I just meant like, I submitted in April 2nd and meeting is October.
Yeah,
And then answer,
right. The RO1s do review, the study sections do meet, I believe, 3 times a year for the RO1s. Okay, thank you all.